Good morning. I guess we should start so that we're not late and give a chance to Dr. Foreman to go through his presentation in timely fashion. So welcome to a new working week and welcome to our grand rounds. So it is my pleasure and honor to introduce our visiting speaker today, Dr. Stuart Foreman. Dr. Foreman is visiting us from Mass General Harvard School of Medicine. He completed his undergrad studies at Wesleyan College, where he majored in chemistry, and then went on to do his MD, PhD studies at Harvard. He did his residency in medical school, his medical school training at Harvard, and then did his PhD at MIT. And he majored in biophysics. Following his MD, PhD education at Harvard, he stayed there to complete his residency in anesthesiology. He did it at Mass General Hospital, followed by a research fellowship where he worked very closely with Dr. Keith Miller, who was a renowned scientist in the field of anesthesia mechanisms. Dr. Foreman started his independent research career after that and has built quite a reputation working on the molecular mechanisms of general anesthesia. His initial focus was on nicotinic receptors, and now he's very focused on mapping out the anesthesia-relevant sites on the GABA receptor. He is a practicing anesthesiologist, very active in the operating room as well. So he's a true clinician scientist in the NIH sense. I got to know Dr. Foreman quite well when we sat together on the NIH study section, surgery, anesthesia, and trauma, where we spent quite a few years working very closely together. I was always impressed with his impartial, very fair, and a very sophisticated review that he would do, helping us not only build a better science, but providing the opportunities for our colleagues to be recognized by the NIH. He will have more of a clinical talk today, and then tomorrow morning at 9 o'clock, he will give a research seminar as well. So if you can attend, please do so. There is a lot to be learned. So without further ado, it is my pleasure to give podium to Dr. Foreman. So help me to welcome him. Good morning.
Thank you very much, Vesna. That was a very kind introduction, and it's a real pleasure to be here on a beautiful morning. This is an awesome space, I gotta say. At Mass General, we used to have an amphitheater where the speaker used to be in the pit, looking up at the audience. So this is like a flashback for me. Not terribly stressful, I would add. So the talk I'm going to give today, I'll just put up this statement because I know it's required, is really a translational talk. It's not really a clinical talk. What I'm trying to convey for you right now is why bother to study general anesthetics and their mechanisms, which is the area of research I've devoted most of my research career to, and part of that is going through the history of the evolution of general anesthetic drugs, which maybe some of you are familiar with, but I can tell you from my perspective as somebody who's working in this translational space now, it's a little bit of an eye-opener to appreciate how the drugs we use every day evolved. And then, so I'll go through that history for the first half of the talk, and then the second half of the talk will basically be about some of my newer research, looking for hopefully future general anesthetics. So let's get started. So general anesthesia, as you know, I mean, you guys take care of, it sounds like, of a significant fraction of these 60 million patients every year in the United States who get a general anesthetic. Of course, many of them are outpatient procedures. And we are dealing with both the pressure from the systems in which we work to be more efficient and to be safer, as well as the fact that we have an aging patient population who come with more comorbidities. And it turns out, as you probably are aware that general anesthetic drugs are the enablers of a lot of surgical technology and minimally invasive, say, radiographic technology and other procedures. So pushing our drugs to perform better and finding better drugs ends up being a really critical factor in limiting how much progress other specialties can make. So as you are aware, no doubt, general anesthesia is a reversible process, we hope, leading to unconsciousness, amnesia, and immobilization. And we have a number of side effects of general anesthesia that you're familiar with. So let's just go through the history of general anesthesia. This is just an overview of inhaled general anesthetics. As you know, these were the first drugs to be discovered, nitrous oxide and ether. The picture you're looking at there is of the Ether Dome, which is at Mass General Hospital. If you ever visit Boston, you can take a tour. It's kind of a cool spot. But one thing to note here is that we haven't had any new inhaled anesthetics since the 1990s. And that's quite a while. And truth is, we may never have a new inhaled general anesthetic. To go through this history in a little more detail, back in the 19th century, general anesthesia was empirically sort of discovered, mostly based on observations, both in animals, but also in humans. There used to be traveling shows where people would inhale nitrous oxide, for instance, and some dentists in the Boston area noticed that these patients, the people who inhaled nitrous oxide got really goofy, but also seemed insensible to pain. And that was sort of how nitrous oxide and ether got introduced into practice. Soon thereafter, other inhalable drugs with these properties were discovered. All of them had significant problems, explosions being a primary one with ether. Nitrous oxide, of course, on its own, insufficient for anesthesia in most patients. Chloroform hepatotoxicity was a huge problem that emerged. Newer drugs came online in the 20th century, cyclopropane, for a very brief period of time, and a truly explosive gas. Fluoxetine, the first synthesized halogenated ether, turned out to also be explosive and had significant toxicity in the kidney. And then, of course, halothane, probably the drug you're most familiar with from our historical past, the first synthesized halogenated alkane, which was non-flammable, and that was one of the main reasons it took over in the operating room from ether, along with the fact that it was marginally faster than ether on induction and recovery. But it had a number of problems as well. And the true evolution of inhaled anesthetics was due to this gentleman, Ross Terrell, who undertook in the 1960s and 70s an endeavor to synthesize a wide variety of halogenated ethers and alkanes and have them tested as potential general anesthetics. And it was based on his work that we ended up getting enflurane, isoflurane, sevoflurane, and desflurane, all of which are, as you know, are ethers. They have low blood solubility and therefore rapid kinetics. They have less problems with arrhythmias than halothane, and they also have far less metabolism and therefore far less halothane hepatitis. The intravenous anesthetic world started a lot later than the inhaled anesthetic world, but probably represents at least our potential future. And you're looking here again at an overview, but we'll go into this in a little more detail. So the ethers, diethyl barbituric acid, the first ether described in the literature was actually first synthesized in 1903. However, and truth be told, thousands of barbiturates have been generated from that initial discovery, all of them having interesting CNS effects. The first intravenous anesthetic with sodium pentobarbital was introduced in 1936 by Ralph Waters. It was, just a revolution in terms of getting patients quickly from awake to anesthetized in comparison to ether, which was the mainstay of inhalational anesthesia at the time. Methotrexate followed soon thereafter in 1960. I don't know how many of you have used these drugs. I used to use pentobarbital when I was a trainee. Methotrexate was widely used at that time too. Don't see them much anymore though. Drugs you haven't heard of, but which sort of made a quick splash and then disappeared, include propranolol. You'll see a little structural analog of propranolol that actually was a drug in development about a decade ago in one of the slides coming up. This was a short-acting intravenous agent, which was formulated because of its high hydrophobicity in a diluent called cremophor-EL. And it turned out that cremophor-EL, it's a bit of a theme of this section of our history, was both painful on injection and tended to cause anaphylactoid and occasional anaphylactic reactions in patients. And so this drug was actually withdrawn pretty soon after it was introduced. Ketamine, a drug you are familiar with, was introduced in the 1970s, but because of other drugs, really never made it as a standalone induction agent. I'm sure you're all using it if you can get your hands on it these days for a variety of other reasons. Alfaxolone, an interesting compound, which is a neurosteroid anesthetic, was introduced in 1971. This was also formulated in cremophor. It was a drug that was also suitable for total intravenous anesthesia, gave relatively smooth inductions, but the cremophor ended up becoming a major problem, and this drug was also withdrawn from the market. Etomidate, a drug we still have on the market, was introduced soon thereafter. There was a lot of excitement about etomidate because of its amazing lack of the usual hypotension and respiratory depression associated with induction by other agents. However, not too long after etomidate was introduced into the market and started to be used quite widely, the problem of adrenal suppression was recognized. And this eventually led to restriction of its use to single dose. And as you know, it's a fairly rarely used. We keep it in our drug lockers because if we get a hypotensive, bleeding patient or somebody with severe congestive heart failure, we will use it on induction occasionally. But truth be told, it doesn't compete with the next drug, which is propofol. And propofol was discovered first in the 1970s. I'll show you the paper in the next slide. And developed, and the key, one of the key developments with propofol was that instead of formulating it in a cremophor, it was formulated in lipids. And that created some problems, but not as many problems as cremophor. And propofol turned out to be a wonderful drug for both induction and total intravenous anesthesia. And it had these nice side effects too, or nice additional effects, I should say, which was that it actually has an antiemetic activity, and patients tend to wake up feeling, if not euphoric, at least pretty normal. So one of the things to appreciate about all of these drugs is very, very few of them were actually, designed as general anesthetics. The activity of etomidate, for instance, as a general anesthetic was discovered when it was just injected into rats. This was an azole drug. It's an imidazole. And it was actually developed by a drug company looking for better antifungal medications. And when they injected it into rats, the rats fell asleep and then woke up again. and they recognize this alternative use. Propofol, we don't actually have a real record of how the drug was discovered. In this paper, the first paper published by John Glenn that describes propofol, he just mentions in a footnote that one of his colleagues observed that diethylphenol caused sedation and hypnosis in rats. And this first paper actually describes, a large number, many dozens of structural analogs of that first alkyl phenol that was discovered, and focuses on propofol as the one that has the best profile for development of a general anesthetic. What they did recognize in the first paper was that propofol could be dosed multiple times, and this is in contrast to barbiturates, I would mention, without having it last longer and longer and longer. So barbiturates, if those of you familiar with the concept of context-sensitive half-life, these were drugs where if you gave an induction dose of thiopental, your patient would wake up in 25, 30 minutes. If you happened to have difficulty with the airway and you needed to give a second dose 5 or 10 minutes after the first dose, your patient wouldn't wake up for an hour. And if, God forbid, you gave a third dose, you'd just park them in the ICU until tomorrow. So there was a significant problem that was recognized with barbiturates, and they really could not be used for total intravenous anesthesia, whereas propofol had a pharmacokinetic profile that actually made it quite attractive. And I should add, Dr. Glenn last year received the Lasker Award, which is pretty close to a Nobel Prize, if you will, for his discovery of this incredibly valuable drug. So the question is, can we do better than propofol? This is another way to word this. And to do that, we can consider from a number of different perspectives. And first is to think about what's the drug you would love to have that you could give to your patient and give them the ideal anesthetic. And of course, safety, we always bring up first. Minimal toxicity, maybe even some organ protection, which is a feature that some of our general anesthetics, particularly the inhaled drugs, seem to bring as an additional benefit. We don't want it to hurt when it's injected. That's one of the downsides. That's why they recognize about propofol and some of the other drugs. We don't want, if it's an inhaled drug, for the patient to be coughing and choking on it. Those of you who have ever tried to induce a patient with desflurane alone have probably encountered a little bit of that kind of reaction. We want it to be quick. We want it to induce anesthesia rapidly, but we also want our patients to wake up fairly quickly after the drug exposure ends and in a predictable manner. We don't want a lot of metabolism so that patients with renal and hepatic disease can tolerate these drugs. We'd like it to be not too expensive to make. Our drug budget is pretty tiny, and we certainly don't want it to become a limiting factor in what we can do for our patients. So one approach, and this is related to my research work for the last 20-odd years, is to try and understand how the anesthetics we use work at the molecular level. So this is a large, busy slide. It represents hundreds of postdoc years of work and many labs working in parallel on a variety of different potential targets. The one target that we feel very confident is actually involved in general anesthesia by several of the drugs you see there are GABA-A receptors. And those of you who are interested in GABA-A receptors, I'll be talking about them tomorrow. And they will be a bit of a theme of this talk, but they, in the last 20 years, have been recognized as the major targets for drugs like etomidate, nalfaxolone, barbiturates, and propofol, although some of those drugs also have secondary targets. So you'll notice, for instance, that etomidate and nalfaxolone have almost no activity at any other of these target ion channels or receptors, except for GABA-A receptors. They are the most selective drugs we have. Propofol, in contrast, although it is a very strong GABA A receptor modulator, has a variety of other targets that it also affects. And of course, the volatile anesthetics, the least potent drugs that we use, which are the volatile anesthetics, nitrous oxide, actually almost every target we look at is affected by these drugs. Why do we look? Well, safety is a priority. It's a very important aspect of what we do. It's why we have our jobs. And you'll see there that the lower number of ion channel targets that were listed on the slide before, the safer these drugs are. So I hope you guys know what a therapeutic index is. It's basically the dose of a drug that kills half the animals in 24 hours divided by the dose that causes hypnosis or loss of, in the case of rats, the typical model, it will be loss of writing reflexes. So it means the ability to put the rat on its back without it immediately turning over and scrambling onto its feet. With etomidate, we can give 25 times that hypnotic dose, and the animals still survive. With propofol, when we get to 2 to 3 times that hypnotic dose, some of the rats stop waking up. You'll notice the drugs in red are the drugs we use every day, right? The least safe drugs, which something to pause and think about. So we're going to move from that framework toward a brief history of the last couple of decades of drug development. I'm going to start with the limited amount of work that's being done in the inhaled anesthetic space, and it is extremely limited, and move on to the intravenous anesthetic space, where there's at least the potential for dramatic improvements. And what you're looking at there is basically just an outline of the next several slides that I'm going to show you. So xenon has been a drug of interest for quite a while. It was actually first discovered in 1951 that inhaling high concentrations of xenon induced a state of general anesthesia. Its mechanisms are thought to be very similar to those of nitrous oxide. But of course, as a noble gas, xenon is totally inert. It doesn't react. It doesn't require any metabolism. It's actually far less soluble in blood than nitrous oxide, and as a result, induction and wake up from xenon anesthesia is significantly faster than from almost any other anesthetic and inhaled anesthetic we give. So studies that have looked at replacing, say, nitrous oxide with xenon, patients actually wake up substantially faster. It has minimal toxicities in terms of cardiac toxicity. As I mentioned, it doesn't matter whether you have kidneys or a liver if you get xenon because it requires no metabolism. It simply leaves the body the same way it came in through the lungs. And it is environmentally benign insofar as it's not a contaminant. I would point out that xenon, however, is a very rare gas. It's less than 1/10,000th of the atmosphere. It has to be distilled from gas, usually deep in mines, and that are somewhat enriched in this gas. And it is extraordinarily expensive. I think the current cost of xenon is over $20 a liter. And that's why and there's basically a very limited supply in the world. So the value of xenon, in order to make this, the use of this drug make sense clinically, is that it should have a substantial positive value for patients. And what that turns out to be is this question of whether xenon is a neuroprotectant. And we've been, and clinical trials have been underway looking at xenon as a neuroprotectant for almost a decade now. There's a tremendous amount of work in animal models demonstrating that xenon can protect the brain and nervous systems of these animals from hypoxic damage, traumatic brain injury, and a variety of other insults. And so it makes total sense that this drug could be the perfect drug for patients having cardiac surgery who are at high risk for postoperative cognitive problems, for patients who come in with head trauma, for patients having neurosurgery, et cetera. I would add that if you read the conclusions on these, I know it's a very busy slide, I apologize for that, but basically the evidence from clinical studies in human beings, and this is sort of a theme in the neuroprotection world, is that xenon, the signal is not that great. It's not a clear signal at all. And so the studies are still ongoing, and I don't think the story will be written about xenon, or the final chapter will be written on xenon maybe in the next 10 years, if we're lucky. The only other inhaled anesthetic that I could find, or that I'm aware of, that had been introduced as a potential drug for clinical development is this compound, which I can't pronounce, but BTTE will do. Dr. Brosnan is a veterinarian who worked with Ted Eager. Does anyone up there know Ted Eager's name and what he's famous for? He's the man who invented Mac, and one of the people who tested all of those drugs that Ross Terrell created back in the 1960s and 70s. So this compound is one of the Terrell products that was never introduced into clinical medicine, but it has biophysical properties that make it a reasonably attractive drug, and it has the molecular property that's of interest is it hits most of the same ion channels as all the other inhaled anesthetics, except for the NMDA receptors. And I'm not sure what the supposed clinical value of that might be, but on the basis of that, this patent was filed a number of years ago. I've never seen a clinical study involving this drug. So we're going to move on to intravenous general anesthetics. And there are a number of strategies that have come out in terms of how we can do better with intravenous anesthetics. And one of the trivial approaches has been to try and improve propofol's formulation in order to get rid of some of the problems, such as pain on injection and limited shelf life. So medium chain lipids are associated with a better shelf life and with a lower concentration of free propofol in the emulsion that's injected, and it's that free propofol that immediately hits the vein that causes the painful response that we observe with propofol injection. Microemulsion simply means making smaller little lipid vesicles. It's a more stable emulsion than the typical the original propofol formulation. Cyclodextrins are those of you who've used Sugamidex, it's a cyclodextrin. It's a barrel-like compound. It's hydrophilic on the outside, hydrophobic on the inside, so we can load cyclodextrins with propofol, and they will release it on injection. The key toxicity that this formulation addresses is if you're giving long-term injections of propofol in the ICU setting, for instance, some patients will get toxicity from the lipid itself. And so here's a formulation with no lipids. However, cyclodextrins themselves can have effects on internal steroid hormones and other compounds. So it's not clear that long-term infusions of cyclodextrins are actually clinically safe. Other reformulated drugs include etomidate in lipid, which reduces the incidence of PONV. So it's an interesting question about whether it's the lipids in propofol that are actually the antiemetic component. It's been looked at by some research, and alfaxolone has actually been reformulated in cyclodextrins as well. It's being reintroduced in the veterinary population. I don't believe there's been some clinical studies in humans, but it hasn't been introduced back into human use yet. This is a drug that I assume the older folks in the room have heard about. This is phos propofol. It's a prodrug, so this drug Again, because of that phosphate group appended to propofol, requires no lipid. It's water soluble, so it gets rid of the lipid problem. To some extent, I believe it reduced pain on injection as well. It does need to be converted to propofol after introduction in the bloodstream, and because of that, it's a bit of a slower onset than propofol. So this drug made it into clinical use. The folks who were selling it thought it might be a great drug for sedation during colonoscopy, for instance. The problem that it had was that phosphate causes an undesirable side effect, perineal burning. And I don't know if any of you have ever witnessed a patient maybe getting a bolus of dexamethasone who complained of a rather warm sensation between their legs. But it is, in fact, quite unpleasant. And as a result of that side effect, this drug really failed in the market, and I don't know if it's even sold anymore. Actually, it's not. This is an approach that has real potential. So soft drugs, you all use remifentanil, you use esmolol. These are drugs that have been engineered based on older drugs like fentanyl and metoprolol to have a metabolizable moiety here, that turns into a carboxylic acid based on nonspecific esterases that are in blood and pretty much every tissue of the body. There is no pharmacogenetic deficiency in esterases in any live human being, it turns out. Remifentanil you can put in patients who have no liver function, who have no kidney function, who have a deficiency in butyrylcholinesterase, and the pharmacokinetics are pretty much unchanged. So these drugs, CPMM stands for cyclopropyl methyl metamidate, a drug that was developed by my colleague Doug Rains with a little bit of our assistance, actually made it into clinical trials. It's not currently under further development, but it has a very predictable short half-life. And it has some of the same advantages as etomidate does in terms of hemodynamic stability and lack of respiratory depression. And I would add that it has lower adrenal toxicity than etomidate as well. Remimazlam is a derivative of midazolam that has the esterase sensitive moiety added on. It's been in clinical development, I think, for almost a decade now. I first heard of this drug more than a decade ago. The folks who own the intellectual property are sort of developing it in a very slow fashion, but I think it has real promise for getting through into clinical use. It's currently in phase 3 trials. And then AZD3034 is actually pretty much the same molecule as propranolol was decades ago. They added a methyl group to make it slightly more hydrophobic, slightly more potent, but you'll notice that it also has an esterase-sensitive moiety. So propranolol and this drug are actually rapidly metabolized hypnotics. And all of these can be used for infusions with essentially zero-order kinetics. The half-life after a couple of hours of infusion will be pretty much the half-life that it was in the first minute. And then there have been several attempts to fix toxicities or reduce toxicities through actual changing of the molecular structure. So the example on the left here is Butyfol, and this is an interesting drug. It was among the compounds that were described in that paper by John Glenn was this sec-butyl, disec-butyl phenol. And in this case, there are actually stereoselective versions of each of those sec-butyl groups. And this was sort of the optimal drug in terms of both lack of pain on injection, potency, and other pharmacokinetics. However, its advantages over propofol, I guess, during clinical trials were deemed insufficient to to be worth pursuing, and this drug, again, no longer in development. Carboetomidate is another drug developed in our group. This was really just a thought experiment. There's a great deal of evidence that the adrenal toxicity of etomidate is based on the interaction of its imidazole, one of the imidazole nitrogens, with the heme group in the target enzyme that's involved in adrenal in its adrenal toxicity. And so by turning that imidazole into a pyrrole, we were actually able to essentially make a drug that has the same shape as etomidate. It still modulates GABA A receptors and causes hypnosis, but it has no adrenal toxicity whatsoever. However, this drug is so hydrophobic that formulation actually turns out to be a problem that the industry hasn't yet cracked, and so putting it into human beings is still not really an option. And then we get into trying to discover new general anesthetics. And this has been going on, I think, for better part of 2 decades now. And most of it, as you noticed when I went through the section on molecular targets, GABA A receptors are essentially recognized as primary targets for some of our most useful, or some of the safest, at least, general anesthetics. And so a fair amount of work has focused on trying to find new ligands that modulate GABA A receptors and produce general anesthesia. The one you're looking at here is a very unusual structure. It acts at the same site as remimazolam and other benzodiazepines, which is somewhat different from the sites where propofol and etomidate, for instance, work. But this is a extremely safe drug. You'll notice its toxicity ratio is 35. That's even better than that for etomidate. It's water soluble. It is reversible with flamazanil. There have been a handful of human beings who have received this drug in Japan. It performed extremely well, and then the drug company stopped developing it about 10 years ago. Don't know why, but I think the market has its own logic. Can we discover new GABA A receptor ligands using scientific processes? So here's the first example that was described in the literature. This came out of Rod Eckenhoff's lab at University of Pennsylvania. It's a very clever idea. So the molecule shown here is horse apoferritin. You can get this from horse blood and purify it. It's a crystallizable protein. You'll notice it has a lot of alpha helical structure. And that alpha helical structure forms what are called 4-helix bundles, such as the one there, which has a propofol bound in it. And that actually mimics the site in GABA A receptors where general anesthetics bind. They combined this protein with a fluorescent drug compound that has anesthetic activity. It's an anthracene analog. But what they found was that when this drug is bound to apoferritin, if they excite the tyrosines and tryptophans in apoferritin, the ligand lights up like a Christmas tree. When it's not bound, it dims the bulbs quite significantly. And so on the basis of that, they created a high-throughput screening technology where apoferritin was bound in these 384 well plates. They added this drug along with a library of other drugs, and the question was, will the other drugs compete for that binding site effectively? If they do, they're potential general anesthetics. And so they could use this as a high-throughput screen. You'll notice that they had a fairly significant hit rate. 16% of the compounds they looked at in a small library actually gave them a positive signal. They used biophysical filters to select a smaller number, none of which turned out to be good at general anesthesia. In fact, the first paper listed here is using that approach with over 350,000 compounds in an NIH compound library and a single compound was identified that I believe was reported to have potential as a clinical drug development base. Haven't heard anything since 2015 on that. The other two papers here describe using in silico search strategies based on structural models of GABA A receptors, and then basically using computer docking of ligands that are in enormous online drug databases to see which fit into the pockets that were identified as anesthetic binding pockets. And a number of compounds have been identified as potential general anesthetics using that approach. Again, at this point, none of them have made it very far along the pipeline. So we're going to switch gears now. So the question really is, are these strategies the right strategies? And a few years ago, we started thinking that maybe they weren't. And part of this is, you may have sort of heard one of the themes of the talk here is that we've been chasing safety and target specificity for the last 30 some odd years in this business. And yet the most popular drugs that we use, propofol, and to be honest, the inhaled drugs, are not highly selective drugs at all. They are, in fact, what we would call dirty drugs. Rod Eckenhoff would call them promiscuous drugs. They're not the safest drugs either. But they have certain properties that make them clinically attractive, and that's why we use them. And we occasionally will choose another drug for a specific patient in a different setting. But for the vast majority of patients, these currently are propofol is the drug of choice. And then many of you out there, I'm sure, are practicing multimodal and intravenous general anesthesia at this point. How many people have used ketofol ever? There you go. So it's a reasonably, mixing ketamine and propofol in a syringe and infusing it into your patient turns out to be a fairly effective approach for many minor procedures. And without a great deal of toxicity, you don't get the toxicity, for instance, of ketamine that you would get if you used it as a drug alone. So on this basis, we decided we would sort of use a different approach, and that would be to go back to the original way that anesthetics were discovered, which is in animals, and actually screen drugs in animals, and do it in a way that created that we could do at a fairly high rate. And so the model we selected, which is a model that's like being used everywhere now, is zebrafish. Zebrafish larvae are tiny little creatures that are only less, 3 or 4 millimeters long. They fit in 96 well plates, and they take up drugs very rapidly. And so we created essentially a high throughput platform for this kind of work. This was not something I did on my own. My main collaborators up there are listed at the bottom. So, as I mentioned, these are vertebrates. They have a rapid life cycle. There are commercially available devices that can do video analysis of their behavior in multiple animals simultaneously. One can make knockout animals to do mechanistic research as well, or knock-in animals. And then there are even further technologies being developed to use these animals for CNS activity, electrophysiology, you name it, calcium spark technology. But this is the assay we developed. It's a very simple one. We put our animals in 96 well plates. This is one of those plates with an animal swimming in it. And they are typically in the dark. And then what we do is we incubate them in the dark for 15 minutes, and then we start a series of exposures to very brief, intense flashes of white light. And the vast majority of these animals, over 90% on the first exposure, will wiggle and swim a little bit after this benign exposure to a flash of white light. So what we're looking at here is their activity, and you'll see there's a spike in activity that then falls away. And we can do that, and then we can expose them to anesthetics while we're doing that. And we can measure two things. We can measure this basal activity in the first 5 seconds, and that gives us a measure of sedation. The triangles here show you the dose-response curve for etomidate as a sedative in these animals. And then we can look at just that period for about a half second during and after the flash of light and ask the question, do they wiggle in response to the light? And that's a measure of what we call hypnosis. Hypnosis being defined as movement in response to benign stimuli. And here you can see that hypnosis requires about 10 times more drug. To suppress hypnosis requires about 10 times more drug than to sedate these animals. This was our first, critical test. We took a panel of known sedative hypnotic drugs and found that the potencies are almost identical in zebrafish as they are to what had been the standard animal model, another small aquatic animal called the tadpole, the Xenopus tadpole. So we can take the data from these 96-well plates. We typically are testing these animals in batches of in batches of 8 to 12 animals at a time at a concentration of 10 micromolar. And so here's the results from one plate looking at the photomotor response, which is the hypnotic test. And you'll see that compound number 85 in our diversity set is a pretty good hypnotic. And this is its structure, which is kind of interesting. In another set here, we found no hypnosis, but we did notice that there is a sedative activity at 10 micromolar for another compound. And we had 2 enantiomers of that compound in the drug library that we were using, which was from colleagues at Boston University. So we can do rapid characterization of these compounds in the animals, looking at their potency for both sedation and hypnosis. This is an experiment that takes 30 or 40 minutes to perform. We also compared them in tadpoles because, again, this was our first set of studies. We wanted to compare to the standard. In terms of the 2 enantiomers that were available, one was clearly more potent than the other in these assays. And we then went further and started looking at their ion channel activities using electrophysiological approaches, relatively simple ones. And for instance, in this case, the first compound we found, number 3237, has activity in GABA receptors, glycine receptors, and neuronal nicotinic receptors among the panel of 7 that we looked at. The other 2 compounds actually showed similar activities in NMDA receptors and neuronal nicotinic receptors. So we're finding compounds. What's exciting to me about these compounds is they're not all GABA receptor modulators. They're potent. They're working in the 10 micromolar range, and so we have a tool here that we believe has the potential for identifying compounds that have the activity we're looking for with some sort of multimodal targeting approach or mechanism. So we've injected these compounds into a limited number of rats. The first compound does cause sedation and hypnosis. So we are currently looking at structural analogs of it to see if we can get a little better potency and get a little better effectiveness in the rodent population. The second set of compounds hasn't looked quite as promising. I will add that we are doing mechanistic research on these as well. For our first transgenic zebrafish model, we decided to knock out the GABA A receptor beta 3 subunit. Those of you who are familiar with the literature on the drugs that work through GABA A receptors, the beta 3 subunit seems to be of particular importance in terms of mediating the endpoints like hypnosis and sedation that we consider important for general anesthesia. So on panel A there is simply, sort of a generic approach to transgenic modification using CRISPR in zebrafish. You can see the specific mutant that we created. It's a 10 base pair insertion into the beta 3 portion of the gene that encodes the extracellular domain. So this is basically what happens is a stop codon is created, and the fish, if they express any of this protein, they're really expressing a non-functional form. You can see that RNA expression drops significantly. That's due to early degradation of the RNA when it's got this early stop codon, and essentially no protein is expressed in the knockout fish. So these fish, we used, and this is data that's currently under review for publication. So the fish are insensitive to the drugs we expected it to be insensitive to, etomidate, propofol, and barbiturates, and basically show normal sensitivity to everything else except ethanol. And the ethanol story is obviously interesting, but at this point, too mysterious for me to even discuss. So I'm sorry to take up so much time, but so just as conclusions, it's important to understand that general anesthetics, by and large, the ones that we use were discovered. They were not drugs that the community decided we needed and that we went out hunting for, except in a few cases, such as that for Ross Terrell being probably the prime example. The current approach, or the most recent approach, is using targeted approaches like GABA-A receptors and hunting for safety don't seem to be working very well. Nothing that has emerged from those research is competing with propofol, and we're hopeful that zebrafish will lead us towards some new compounds that may have more value in the clinical setting and maybe enable more developments in the future. So thank you very much. I should give credit to all the people who helped with the research I just discussed. It's a lengthy list, so I'll just leave this slide up and take any questions. Yes?
I'm just curious, are knockouts for any of the receptor types?
Say again?
Are knockouts for any of the receptor types lethal?
Great question. I can tell you that we are working on one of the other GABA receptor subunit knockouts, or have been working on it for about a year, without much success. And we may end up concluding that particular knockout is in fact lethal. So I can't really discuss it with you at this point. But some of the other knockouts that we've made have phenotypes that are also unexpected in terms of their behavior, which creates other issues as well.
Have you tried so far to see whether there is correlation with substances separately in zebrafish to inject it in mice?
So we haven't really looked at enough. You mean, does the potency in a zebrafish predict potency in the mouse? One would hope so, but I think there's a lot of experience with the benzodiazepines in particular that would suggest that is not necessarily the case, and a lot of that has to do with the protein binding and limitations on crossing the blood-brain barrier. So some of the most potent benzodiazepines in molecular studies turned out to have lower potency in the animal models because the protein binding was so high. If 99.9% of the drug is protein bound, then only a tiny fraction is in the form that can cross the blood-brain barrier. And it creates all sorts of pharmacokinetic problems. So we don't know is the starting answer. But there are strategies, molecular strategies to try and, if you find activity, to try and alter the structures so that you can fix some of these problems.
So what is the average price to develop it for a study? What do you say?
So the question is, how much does it cost to develop a new general anesthetic? One of the issues in the whole field of developing new general anesthetics is that the market for general anesthesia is pretty small when you compare it to the market for drugs that people take on a daily basis, or some of the new, biologicals and things like that. And so only drugs that can be developed cheaply end up getting developed at all. And I would say, we're talking about $100 million on the outside would probably be what. And I would also point out that in the drug development sort of environment, it's usually medium to small companies that are interested in these drugs rather than big pharma. And again, that's all because of the potential market is so small. These drugs can have a significant impact if your capitalization is just a few $100 million. But if you're a $100 billion drug company, then these drugs are nothing. Thank you all for your attention. Have a great day.